Hello everyone, I am Abdul Muiz Ahmed, and in this video we will be resuming off with the second chapter of our textbook, that is personal identity. And now we would be covering the fourth part of this chapter, that is identification of sex of a living person. Basically, identification of sex involves three aspects. Firstly, there is identification of sex of a normal living individual and identification of sex in doubtful cases. Secondly, there are certain intersex states or bisexuality that need to be understood. And lastly, we need to learn how we can identify sex of a dead person, like in any form of dead body, whether it's mutilated or decomposed body, or in the form of fragmentary remains as well. We would be covering the first aspect in this video, so let's just quickly jump to the learning objectives. At the end of this lecture, you will be able to: number 1, describe the three major types of evidence on which sex can be determined in a normal living individual. These evidence include the ones that are more certain, the ones that are highly probable, and the ones that are presumptive. Number 2, you will be able to name the methods which are used to determine sex in doubtful cases. Number 3, you would be able to explain nuclear sexing with a special reference to the Barr body and Davidson body. Number 4, apply concept of study of sex chromosomes to identification of sex of an individual. And lastly, number 5, describe medical legal aspects and importance of sex determination. Alright, so let's start with our first learning objective, that is to explain the three major types of evidence that we can use to establish the sex of a normal living person. The first type of evidence are the ones that are more certain, meaning that if these evidences are found in an individual, they give around 90 to 95% guarantee regarding the sex of an individual. So let's take an example. If a normal living individual is brought to you, and you as a medical legal officer have to establish the sex of that individual, so you do the examination, and in the examination you find that there are functioning testes, and these are functioning because they lead to emission of seminal fluid. So in this case, that individual would most certainly be a male. This is what you will say. On the other hand, if you find functioning ovaries instead, and by functioning I mean presence of menstruation at puberty, so in this case that individual would most certainly be a female. And secondly, in prepubertal cases, you can run a chromatin pattern test instead, and you can demonstrate the sex chromosome pattern. If it's XY, then most certainly that individual would be a male, and if it's XX, then most certainly that individual would be a female. As far as your examinations are concerned, from exam point of view, you only need to remember about the functioning testes and functioning ovaries as the evidences that are most certain, that are most certain to establish the sex of a normal living person. So if someone asks you, how can you determine the sex of a living person? In the most certain way. So you can tell them by checking the gonads. You won't say that you can do it on the basis of their clothes, or on the basis of their appearance, or on the basis of their external genitalia. Instead, you would say that you will check the gonads of the individual, and you would provide the most certain evidence regarding the sex of a normal living person. Moving on to the second type of evidence, and these are the ones that are highly probable, meaning they give you an above average idea regarding the sex of a normal living individual. So let's just take an example again. If an individual is brought to you and you have to establish the sex of that individual, so first thing first, you are going to check for gonads to establish the sex in the most certain way. But if you don't find any functioning testes or any functioning ovaries, in that individual. So you are going to move on to the second category of evidence that are highly probable ones. So you check for external genitalia. Now, internal genitalia, if you remember, were our first category of evidence, that are the ones that are more certain. And external genitalia and breast tissue are our second category of evidence, that are the ones that are highly probable. So if in that individual that we took in our case, you find penis, you find prostate, you find seminal vesicles, and upon examination of breast tissue, you find no breast tissue at all, so you would say that that individual is highly probably a male. Alright, on the other hand, if you find vagina instead, you find uterus, you find fallopian tubes, you find that that individual has well developed breast tissue, has well developed breast, so you would say that that individual is highly probably, again saying highly probably, a female. Okay, so just to summarize, first category of evidence, the more certain ones, and basis is internal genitalia. And then second category of evidence, the highly probable ones, and basis is the external genitalia and breast tissue. Now the last and third category of evidence that we have got are the presumptive ones. This class of evidence includes noting down the outward appearance of the individual, looking at the features and general contours of the face, observing the presence or distribution of hair, and looking for evidence of shaving. You can also observe the scalp hair and check out their clothes, their habits, their inclination, and voice pattern, etc. etc. So these are the features that you are going to look for and record them as presumptive evidence. Now let's compare these features in males versus females. Number 1. If we talk about the general build and muscles, so males have larger build and are more muscular as compared to females who have a smaller build and are lesser muscular. Number 2. Males have broader pectoral region than pelvic region. Pectoral region, as you might remember from your second year, is the one that is comprising of shoulders and chest, and pelvic region is the one which is comprising of hips, buttocks, and thighs. So males generally have a broader chest region, that is pectoral region, as compared to their hip region, which is their pelvic region. While in case of females, they have a broader pelvic region. They have a broader pelvic region, that is their hip region is broader, and buttocks as compared to their chest, and moving on to number 3 now. Number 3, in males the waist is ill defined, while in females their waist is well defined. Number 4, males have buttocks that are flatter and contracted, while in case of females the buttocks are full and rounded. Number 5, males as well as females have a particular specific distribution of body hair. In males, the hairs are thick and coarse and extend from pubis to abdomen, and there are hairs over the entire surface of chest and on the face that appear after puberty. While in case of females, the hairs are thin, fine, and are covering mons only, and there are no hairs on the chest and on the face as well. Number 8. Males have scalp hairs that are shorter, thicker, and coarser, in contrast to scalp hairs of females that are longer, thinner, and finer. Lastly, males have Adam's apple or pomum adami or larynx that is very well prominent in the middle of the neck, and in females they don't have prominent Adam's apple or pomum adami or larynx. So these are the general features you observe or look for in an individual and call them as presumptive evidence, or record them as presumptive evidence for establishing the sex of a normal living individual. So just to summarize, we have got 3 categories of evidence that we can record in order to establish the sex of a normal living individual. Number 1, first category are the most certain ones, and they include internal genitalia. You have to check for internal genitalia, that is testes or ovaries. Second category are the highly probable ones and include checking external genitalia. along with breast tissue. And lastly, third category are the presumptive ones, which include recording the general appearance or general outlook of an individual. These three evidences are used to establish the sex of a normal living individual. This covers this covers our first learning objective. I hope all of this makes sense till now. Now let's move on to our second learning objective, which deals with the methods of determination of sex in doubtful cases. So in such cases, true sex of an individual can be determined by following 5 steps, which are: number 1, doing a thorough external examination; number 2, doing a thorough internal examination; number 3, performing gonadal biopsy; number 4, performing nuclear sexing; and number 5, studying sex chromosomes. We are going to cover the last two ones in detail, so let's go through nuclear sexing first. Nuclear sexing. What do you mean by that? You need to understand its definition first. So nuclear means nucleus, as you all know, and sexing means to assign sex. Hence, you would be assigning sex by looking for something specifically in the nucleus of cells. Alright, this method comes into play when determining sex in doubtful cases, in cases of decomposed bodies, in cases of mutilated bodies, and fragmentary remains as well. The principle behind this method is based on the difference in the nuclear chromatin pattern between male and female cells. There are slight differences in the chromatin pattern that you observe and note down, and then determine whether the cells are from a male individual or are from a female individual. Now, what are these differences that you look for? They are 2 in number and include A, Barr body, and B, Davidson body. So these are the 2 differences that you look for. We are going to study each of them now. So firstly, Barr body. Barr body, as most of you would already know, is a small plano-convex or pyramidal mass of chromatin present at the edge of nuclear membrane. I will show you a picture of that in the next slide as well. Now, if the cells contain Barr body, then they are stated to be chromatin positive, or if not, then they are stated to be chromatin negative. As for the stats, in a buccal smear, the percentage of cells showing the presence of such Barr bodies varies from about 20 to 80% in females and 0 to 4% in males. So, for example, in females, if you take 100 cells from a female, so around 20 to 80 cells would be chromatin positive. And in males, if you take 100 cells, so around 0 to 4 of them would be chromatin positive, and 90% or more of them would be chromatin negative. Finally, the tissues. About the tissues that are used to identify Barr bodies are those that resist autolytic changes. In forensic medicine, for evidences, you always try to take those tissues that resist autolytic changes, that resist putrefaction, that don't decompose and remain alive and remain there for large periods of time. Examples include root sheet cells of human capital hairs, buccal scrapings, cartilages, etc. etc. So now moving forward to the diagram. This is a diagram which is showing Barr body. As you can recall its definition, that it's a small plano-convex or pyramidal mass that is present, mass of chromatin material that is present at the edge of nuclear membrane. Okay, so next in line we have is Davidson body. So what is that? It is a thin stalked drumstick-like projection on the polymorph nucleus. This is very important to remember. On the polymorph nucleus in about 6% of cells, especially WBCs of feminine trait, of females. Okay, I have added an illustration of it as well. This drumstick-like projection is Davidson body, and just to compare, this mass at the edge is Barr body. So this sums up nuclear sexing. It is, as you can say, is a way to assign sex by looking at nuclear or chromatin features, we have to look for two of them. One is Barr body, which is a small plano-convex or pyramidal mass of chromatin present at the edge of nuclear membrane. If it's present, then cells are called chromatin positive. If it's not present, then cells are called as chromatin negative. It is majorly present in females, and males are mostly chromatin negative. And lastly, the best tissue that is used to check for Barr body is root sheath cells of human capital hairs. And the second body that you have to look for is the Barr body, which is a thin stalked drumstick-like projection on the polymorph nucleus of the cells of feminine trait. Especially they are present in WBCs. Moving on to the next and last method that is used for identification of sex in doubtful cases, and that is study of sex chromosomes. You already know from your basic understanding of biology that there are two types of sex chromosomes, an X chromosome and Y chromosome. So in this method, you demonstrate sex chromosome pattern in the dividing cells of the body. Examples of dividing cells include blood stains, cartilage, bone marrow, teeth pulp, and hair. One way to do this is to do karyotyping. You can do karyotyping and then determine whether the cell coming from a certain individual is having XX sex chromosome pattern or XY sex chromosome pattern. Alright. Other methods you could do is to do a fluorescent test, and Y chromosome in particular in male is fluorescent to quinacrine. And finally, root sheath cell of human capital hairs are of special interest in this case as well, because of the three distinct characteristics. Number one, as I mentioned you, they resist putrefaction, so they can be used for a long period of time. Number two, Barr body can be demonstrated in it. And number three, Y chromosome as well can be demonstrated in them. Why can't we demonstrate Davidson body in it? Because as I have mentioned you earlier, that Davidson body is only present on polymorph nucleus of WBCs. So now I hope you have a clear understanding about the different ways that you can use to determine sex in doubtful cases. There were 5 in number as I recall. Number 1, do a thorough external examination. Number 2, do a thorough internal examination. Number 3, gonadal biopsy. Number 4, nuclear sexing. And number 5, study sex chromosomes. Now, our last learning objective, which raises the why question. Like, why we are determining the sex in the first place. We have learned so much about the methodologies, and we will be learning more in the upcoming lectures as well. Methodologies regarding sex identification. But why are we learning this? Why is nuclear sexing done? Or why are we looking for evidence or doing karyotyping? So basically the answer to all these lie in the certain medico-legal aspects regarding sex identification. We determine sex for number 1 just for complete identification of living or death. Number 2, there are certain civil rights that are reserved to one sex only, like heirship. Males get 2/3 of inheritance, as you know, while females get 1/3 of it. So, and other civil rights include those of marriage, military services, etc. etc. Number 3, sex identification is important in cases of impotency, rape, legitimacy, divorce, paternity, etc. And lastly, it is necessary for simple identification of dead individuals that are in a state of decay, that is where primary organs have decomposed. So you can run a nuclear sexing test, sex chromosome pattern test in these individuals, in these cases. This sums up our lecture. Just to recall, we initially covered the evidence that are used to establish sex of a normal, I repeat, normal living individual. These were 3 in number and were called as number 1, most certain ones, the basis of which is internal genitalia. Number 2, highly probable ones, the basis of which is external genitalia. And number 3, presumptive ones, which depend upon general appearance or outlook of an individual. Then we learned about the 5 different methods that are used to identify sex in doubtful cases. We studied two of these methods particularly in detail. These were number 1, doing nuclear sexing, and number 2, studying sex chromosomes. Nuclear sexing involved looking for two bodies inside the nucleus, which were number 1, Barr body, which as you remember was a small plano-convex or pyramidal mass that is present at the edge of nuclear membrane, and Davidson body, which is a thin stalked drumstick-like projection present in the polymorph nucleus of cells of feminine trait. Lastly, we covered the major medical legal aspects pertaining to sex determination. If you have any queries or suggestions, you can drop that down in the comment box. If you like this video, then don't forget to press that thumbs up button and share this with your fellows too. Lastly, don't forget to subscribe and follow us on our Instagram page. Thank you so much for listening. Keep learning. Take care. Goodbye.